You're on.
Good morning. My name is Henry Gibbons, and I'm a research microbiologist at the U.S. Army Combat Capabilities Development Command Chem Bio Center. And I want to thank you all for coming out and joining in on this seminar. It's a great opportunity for me. I've been really looking forward to this. And so let me kick in my screen share here, and we'll get started. So the title of my talk is "Whose Spore Is It Anyway?" And it's so I've made a little bit of a tweak to the title that's in the program, but it says "Adventures in Biodefense Microbiology from the U.S. Army." So what I'm going to do today is give you an overview of some of our work, some of the work in my laboratory and group, and I got to start this out with an obligatory disclaimer because I work, because of who I work for, and they're my opinions basically. Some of them are my opinions, except the data. They're data, but all the work here has been cleared for public release. So the talk this morning is divided into 3 parts. I'm really going to give a brief history of sort of biological warfare and biological weapons through history to start with. I'll give you a little bit of overview of the Combat Capabilities Development Command Chem Bio Center. And then we'll have a little bit of a chance to dive into some science and some of the microbial forensics that we've done on an old BW surrogate strain. We're going to give an introduction to testing by PCR. I put that in quotes for a reason. And then we'll talk a little bit about some of the synthetic biology work that we have done for biodefense purposes. And those of you who are going to go to the synthetic biology panel later will have a chance. We'll get a little bit more of an introduction. But what discussion of, or what science fiction fantasy convention would be complete without a mention of he who shall not be named? But you can see I'm drawing a deliberate analogy of the dark arts in the Harry Potter universe to the dark arts in the modern technology, chemical and biological weapons. On the top slide, you can see a picture of the chemical plumes raised by German soldiers or the German forces across the battlefields of Ypres in Belgium. And those are sort of let go and blown downwind in advance of the formations of troops coming afterwards. The intent, of course, was to disrupt military operations in that space. And then later, in the same round, in the biological space, what you see here is a picture taken on a fully protected guys and people in full PPE on Grunard Island, which was the British anthrax testing ground. It was off the coast of one of the British Isles, and this area remained contaminated. So what you can see here is that there is a real potential for forces that can be used for good purposes or for beneficial purposes for humankind to be turned to a much more malignant purposes. This is not just true of chem biospace. It's also true of, we saw that in the nuclear realm. We've seen it in other areas of technology as well. And I'll get into a little bit of that. Oops, why is my screen? Okay, there we go. Okay, so I'm really, for biological purposes, I'm going to split the dark arts out into 3 major categories, one of which is newer. But you have 2 basic components: biological warfare, which is the weapons used in armed conflict; bioterrorism, which is the use of these biological agents or toxins for purposes of sowing disruption for political ends, usually by non-state actors. Sowing fear, mass panic, and that sort of thing. And then newly emerging is this phenomenon of biohacking, or this idea of biohacking. And I describe it sort of as the dark arts face of the DIY bio movement. You can see here this website from a number of years ago. I'm not sure if it's still up, but it came out. You can read a hackerspace for biotech. Experiment with friends. What could possibly go wrong in something like this? Fortunately, these communities have generally engaged very well with the scientific community. They have subscribed to ethical, accepted ethical standards for experimentation on human subjects. But obviously the potential for that is that things go wrong, that people don't subscribe to those standard ethical oversight procedures and start to go rogue. So, for a couple of histories and thought, on these fields, I encourage you to go to some good, reasonably good publications. These are books that are out there available. Judith Miller's Germs, which is the description of the United States program of old, and the exhaustive treatise by Milton Leitenberg and Raymond Zelinskas on the Soviet program, which is it's a long read, but it's exquisitely referenced and very, very well documented. So there's a long history of this, as there are for many techniques in warfare, and it helps to remember that infectious diseases, up until the 1940s, with the advent of penicillin and antibiotics, was the leading killer of soldiers, far and away bigger than battlefield casualties. An infection was often lethal. So soldiers have been aware of the potential for disease to complicate life dramatically for a very, very, very long time. But in 1348, there are some documented some writings that indicate that during the siege of Kaffa, which is over here in the Crimea, it was a Genoese trading outpost out here. It was besieged, and the invading armies catapulted, or thought to have catapulted, bodies of infected people and animals across the city walls. And then this then happened coincident, or perhaps because of this, plague then hopped onto the trading routes on the ships, spread into Western Europe, and then proceeded to kill off 25 to 50% of the population. So it's not clear whether this actually occurred, whether this actually resulted in the Black Death spreading to Europe, this attempt, but it certainly suggests that there was an attempt to do so. A more clear-cut attempt to do this, or at least encourage it, was the advocacy by Lord Jeffrey Amherst, who was the colonial governor in the United States at the time, and he's shown here. He's the namesake for a number of towns and at least one, and one college, which happens to be my alma mater. But he advocated in a truly horrid letter that smallpox-infected blankets be distributed to the Native American tribes who were engaged in insurgency at the time. And obviously, during that colonial era, there was no prescription, or there was no proscription against use of biological agents in warfare. So this is kind of horrific, but emblematic of the colonial attitudes at the time. But things really got going in the 1800s with the discovery of the germ theory of disease, which was really the seminal concept in microbiology. Discovered bacteria and viruses later could actually cause disease. And this is Robert Koch and Louis Pasteur, both of whom worked on Bacillus anthracis, or anthrax, which was an agricultural problem at the time. And then in World War I, chemical warfare really came into its own. The first use at the Battle of Ypres, which I showed, chlorine gas, and then phosgene mustard agents came along. Concurrently, there were attempts to use this new germ theory, these new germs, to sabotage, in acts of sabotage. There were instances where German spies attempted to infect horses transiting the Atlantic to supply the Allied forces with glanders, which is a disease caused by Burkholderia or Burkholderia mallei. So, and obviously this was an attempt to compromise the Allied war effort. But in 1925, the League of Nations, and among others, established the Geneva Protocol, which prohibits the use of biological and chemical weapons, considering that these to be unethical and sort of ick factor weapons in war, and were not an honorable way of doing business in warfare. However, that didn't stop the development of these weapons. In particular, in the 1930s, the Japanese Army and Empire established units, the infamous Unit 731, out in Harbin, China. this then led to the deployment of plague-infected fleas against cities of some Chinese cities, and also the extensive human trials on prisoners and some pretty horrific human rights violations that occurred during that time. The British then fired up a program in 1939. They constructed the research facilities and the development facilities at Porton Down. Their initial work was anthrax, botulinum toxin, and that sort of thing. It was led by just this gentleman here, Paul Fildes, who was a very well-known microbiologist. Then in 1942, the United States got into the war, and Roosevelt tapped George Merck, who then tapped Ira Baldwin, who was a professor at the University of Wisconsin, a bacteriologist. And keep Wisconsin in mind because we're going to come back to it. But he was a professor of bacteriology, very well known at the University of Wisconsin, and interesting. He has an interesting quote here, and he was a Quaker by upbringing, and he is quoted as saying, "You start out with the idea in war of killing people, and that to me is the immoral part of it. It doesn't make much difference how you kill them." That is, I mean, that's an example of the kinds of ethical calculations that people have engaged in order to justify working in this space. So the United States continues work after the war. In 1945, we captured and debrief Shiro Ishii, take some of his knowledge in the dark arts, and we start up our own rather extensive R&D program. And then in 1972, Richard Nixon, effectively by executive order, ends any offensive work that was occurring at Fort Detrick and other places. And then in 1973, the United States signed on, along with 165 other nations, and as of 2014, to the Biological and Toxin Weapons Convention, which prohibits offensive use of development and stockpiling of biological weapons and toxins. So the concurrent with that, a large ramp up in the activity in the Soviet Union occurred, and this was, a covert program. It was huge. It was a large program. It employed at its height about 50,000 people. There were many, many laboratories and institutions. You can see a map scattered of these facilities that were scattered across the Soviet Union. They weaponized, produced, and developed any number of agents. They did open air testing at Vojvodenia Island, which I might add today is no longer an island because of the lowering of the Aral Sea. And some of these facilities are shown here. This is a fermentation facility that's used to grow, Bacillus anthracis. And each of these tanks, each of these dome structures here represent the top of a fermentation vessel. Each of these is approximately, I think there's something around 60,000 liters or 15,000 gallons. Our biggest plant, I believe, at its top stage was in Terre Haute, Indiana. It was never able to really contain the aerosols, and so the United States government, the Army, sold it off to Pfizer, who then used it to make streptomycin for the next 30 years. So I just want to point out here that these technologies have significant use in the civilian sector as well for benign purposes, and for actually medical purposes. So so keep that in mind when you're thinking about these kinds of agents. So some of the fallout from the Soviet program in 1991, obviously the Soviet Union collapses, and some of the knowledge just spills out. A couple of some defectors come across Pasechnik and Alibek, and then there are others who are recruited and emigrated to the United States. But they have all given details on how this worked and how this program was organized. One of the popular books to this effect is Ken Alibek's information dump called Biohazard. It's sensationalized, but it is still a pretty scary read, all told. Some smaller programs in different nation states, in Iraq, I think apartheid era South Africa had something along those lines. But largely the work that's been done in countries, in US and NATO countries, China, Russia, has been defensive. And the idea is that you want to be prepared if somebody decides to use it against you, but no, you don't want to develop it yourself. There's too much blowback. Bioterrorism, on the other hand, has been an active thing. There have been three major uses of or attempts at bioterrorism. One, this guy up here, is the Bhagwan Sri Rajneesh, whose followers in Oregon infected a number of salad bars in his area in an attempt to influence the results of a local election. Talk about an election interference. Here you go. So here, this is they used a common lab strain of Salmonella, which I actually ended up using in my graduate work. The anthrax attacks on Tokyo was an attempt by the cult Aum Shinrikyo, who also did the sarin attacks in the subway, the chemical gas attacks in the subway there. They also attempted to hose Tokyo down with anthrax spores. Unsuccessfully, because they used the vaccine strain inadvertently, whether by deliberate sabotage or not, it's not clear. And then in 2001, the anthrax attacks on the Postal Service of the United States occurred. You can see here in one of the infamous letters, this one addressed to Senator Tom Daschle. There were others that went to other prominent figures. In all, it killed seven people and sickened about 22, 23 others, and cost about $2 to $3 billion to remediate the facilities. So a very significant impact. And it launched a lot of what we now know of as the biodefense field. So where do we fit into this? I'm going to give you a quick overview of the CCDC Chem Bio Center. For those of you who know the history a little bit, we used to be known as the Edgewood Chemical Biological Center, and before that, the Edgewood Arsenal. And so the main mission of that is, what do we do? We're about 1,400 people, a mix of civilian and military. Our mission is to develop and research, development, and engineer new and effective chemical and biological defense solutions. So we're really in the business of innovating in such a way that we can be better prepared to deal with chemical and biological attacks against the soldiers of the United States, soldiers and military folks of the United States. So we've got a number of, we have 4 facilities in different states. Aberdeen Proving Ground, Maryland, that's where I'm based, is just east of Baltimore, Pine Bluff, Arkansas, Rock Island, Illinois, and Dugway Proving Ground out in Utah. And so that gives you an idea. We're a lot of scientists and engineers and some technicians and specialists and deployable folks. Another major activity that we do is we actually go out, we have deployable teams that go out and clean up old chemical rounds from World War II and other eras like that. And whenever somebody finds an old ammo dump, our people have to go in there and use specialized equipment to clean it up. The major missions, you may have heard about Fort Detrick and the work that's done there. They do the medical side of biological defense, countermeasures, drugs, vaccines, and the like. We do the detection, the environmental end of things, outside of the skin, detection, protection, decontamination. So you can see a guy, a soldier here in chemical and biological protective gear. We've have hands in the masks and the development of that. And in new ways of developing decontamination methods. We're a fairly good sized laboratory, about 1 million square feet of laboratory space, BSL-1, BSL-2, biosafety 3 containment laboratories, some of which are also capable of handling explosives. We have chemical surety hoods for the chemical protection agent or mission. And we're involved in a number of new and interesting ways of trying to develop decontamination and detection methods. We're using a lot of synthetic biology techniques to develop chemical sensors and biological and sensors for biology or biological agents. We study the agents themselves so that we can develop better risk models to predict where things go and how things work. And we're trying to develop cellular toxicology methods to help get away from traditional animal models for toxicology. Reasoning, if you can go into in vitro systems, you can use fewer animals and have better results. So now you can give it a sense of who we are and what our mission is. We're gonna dive into the science a little bit. And I'm gonna tell you a couple of stories. They're more or less related. They all have to do with bacillus spores. The first of it is really sort of a microbial forensics when we dug into the history of a surrogate strain that was used in place of anthrax as a safe alternative. So first, let's go into the background a little bit here. I'll tell you a little bit about anthrax and its life cycle. Anthrax has been particularly attractive to nations that have decided to go down the route of developing biological warfare, because it exists in a large part of the life cycle in a dormant spore form, which is really, really hardy. And this spore form, you can see it's multiple layers. It has excluded most of the water from its cells. It's dormant. It's packed its DNA very tightly and protected itself. And it can sit in the soil for decades and survive, which also means that you can do many, many things to it and try to kill it, and it will still remain viable. Typically in nature, this is a disease of cattle and other animals like that. They will ingest or inhale the spores while feeding. That then gets into the lungs or into the bloodstream, where it then causes massive sepsis and bacteremia, growth of very high titers in the blood, kills the animal. The animal then bleeds out into the soil, where the bacteria are released, where they have a decision whether they either survive in the soil if there's nutrients there, or they, upon starvation, sensing that they're starving, they then sporulate and sit around and repeat the life cycle. So a diagram of the spore is shown here. You can see the multiple layers in this, the protective layers on the outside of the spore coat. So I talked about surrogates and why we do that. Well, in order to have an effective defense, you got to understand how the wild organism is actually going to behave. And so you adopt something that you can use either in the field or in the laboratory in order to understand the behavior of the virulent organism without causing risk to your humans or the people who are doing the tests, or to animals in the area, or to plants. So you're looking for something that is similar in biological, chemical, physical properties that is safe to use or minimizes risk. So when I first started, we were asked to fire up a genomic sequencing laboratory that would be quickly able to come out and respond to a potential biological attack, characterize the agent, get a full nucleotide sequence of it, and start to be able to predict how these things are going to behave and make an informed assessment on it. And we had just turned on the laboratory. We were looking for something to throw onto the sequencer to prove that we could do this. And so we chose the old surrogate, which was Bacillus ATROPHAEUS or Bacillus globigii. And you can see that the taxonomic names of this are kind of a mess. These are all the same organisms that just have been known by different names over the years. We put this on there, and we realized very quickly that, yeah, this is not the lab strain of Bacillus subtilis. It's something very different. It's not anthrax either, which was also good news, obviously. But I then made a call to my predecessor and told him what we were doing, and my predecessor in this position, and I told him what we were doing, and he said, "Wow, that's really cool. I know a guy at the University of Wisconsin who has some old strains, some old spore stocks." And so I called up John Lindquist, who is the teacher of the bacteriology labs there, and we got to talking about this. Yeah, I got this great spore stock, it's an old paper, and you can see the old labels on it. Then these labels unambiguously indicate that it came from Camp Dietrich at the time, and even gives a name of a person who had provided it. And they also had some original material that presumably went out there. So we plated these out, realized they had a very different appearances, some of them on this thing. We also dug in a number of other strain collections. And then, but the fact that we were able to see different morphologies here, and you can see 3 different morphologies that came out of this tube here. And this reminded us of what was going on in the investigation, where it had just been published in the investigation of the anthrax attacks. And so these were 5 morphologies. They're different colony types. They're the same organism, but they just have they look different on agar plates. And this combination of morphs and the genomic sequences attached to them was what allowed the FBI to then start narrowing down where this material had come from. And eventually they attributed it to a single flask that was stored at Camp Dietrich, or Fort Dietrich at the time, actually. So we started looking at this. And so we said, okay, let's sequence everything. Let's just do it. Let's do them all. We got a sponsor to buy off on this too. So we did this, and sure enough, we have 3 Dietrich isolates here. We've done each of these. I should mention that this 1942 strain, which is back here, this one. That served as our control, our reference. And we then mapped the entire data set. This is all 4 million or so base pairs on there. We mapped the entire sequence onto that. And anything in black here indicates a mutation or a change in the nucleotide sequence. Not all of these are informative or interesting. Those are, there are some that are synonymous, which don't change anything. But the ones that are indicated by N here are potentially interesting. And in that they changed a protein sequence that is encoded by that gene. And so we then started looking at this, and one of them jumped out to us in particular because it was present in, this mutation was present in most of these military isolates that had some pedigree with the military lineage and some provenance through that. But absent in this and absent in our wild type strain that presumably is the progenitor for this. So that indicated that, okay, something interesting happened here. And we started looking at what this gene is. It's in a gene called SPO0F. Well, what does that do? So you started looking around and checked up on things. SPO0F is really the central integrating node for the decision on whether these bacteria go into the sporulation process. SPO0F integrates environmental signals that come through kinases or proteins at the cell wall that function as sensors. It integrates these and these each add a phosphate group to SPO0F, which then adds a phosphate group to SPO0B, which then adds a phosphate group to SPO0A. And in so doing, we amplify the signal. And ultimately SPO0A activates a DNA binding activity that results then in the transcription of all the sporulation genes that trigger the subsequent differentiation of the bacteria into spores, into endospores. So this really is a critical piece. Timing is critical in sporulation, and it's under strong selective pressure. What you see here is an alignment of an excerpt of this SPO0F sequence to all of the other SPO0F proteins that were available to us at the time. And you note that this H position here, which was present in the 1942 strain, is changed to an R. This is a big change. And we note that with one exception down here, and more or less in some halophilic organism in some salt lake, I can't remember exactly where this was found, but this is universally conserved, and it's a highly invariant residue. So this suggested some strong indication of functions. We started asking ourselves, what could it be doing? Well, we thought, okay, sporulation, that's interesting. What would the effect be on sporulation? And sure enough, when we did this, we compared the Dietrich 1 here, which has the wild type allele, and the Dietrich 2 strain, which has the SPO0F allele, the 101R allele. We noticed that yes, indeed, these sporulate at much higher frequencies than the wild type after 48 hours. And so we started digging in the literature, and sure enough, they actually deliberately selected one of these mutants to be the surrogate strain that's actually used. Over here, you can actually see our own facility here at Aberdeen Proving Ground. This is a 1,000-liter fermenter here. And you can get a sense of the scale at which this, and why having something that sporulate highly in this circumstance would be of interest from practical use. We then did we profiled with this on a high throughput phenotyping scheme. And this is basically 1,500 different growth conditions, all run in parallel on a single incubator. It was a big experiment done by our colleagues at the Naval Medical Research Center. And one of the things we were looking for, what could these other mutations be involved in? One of the things that jumped out at us is that when you compare this to the 1942 strain, which is not shown here because it would be all black, green represents better growth and red represents worse growth in this heat map. And these all are media containing higher amounts of sodium lactate or lactic acid. Well, why would this be important? Well, it turns out that in that same paper, we found out that they were selecting deliberately selecting a sporulating mutant. They were growing it in corn steep liquor, which is a cheap medium that's high in protein, sugar, and, you guessed it, lactic acid. So another adaptation. So really, to sum up those of works, we have two adaptation events, one of which is probably was inadvertent here, where it was artificially adapted to this cheap artificial medium. And the second was basically a case of, ooh, I want that one, which is the selection and propagation through that military lineage shown up here of that hypersporulating mutant. So big teams of people involved in this. This gives you the idea of where we are, of the group at Edgewood that did the work. Bioinformatics crew was not at all trivial in this. NMRC, the Naval Medical Research Center folks did the high throughput phenotyping. University of Wisconsin obviously provided materials. There's big teams of people, and the sponsors, of course, the TMTI program through the Defense Threat Reduction Agency. And some work I didn't talk about today that basically verified the sporulation phenotype and how it could arise. So I'm going to switch gears here, and I'm going to jump a little bit into synthetic biology in the time I have remaining, and some of the work we've done on that. It's an interdisciplinary area that basically seeks to use biological parts as one would Legos. And so we want to build a lot of different things, fabricate things that weren't in existence before, and combine chemical synthesis of DNA with the knowledge of this. So things that don't exist in the natural world, repurposing of biological components for purposes that benefit us. And the first work we did was to make synthetic genetic barcodes for a DoD surrogate. Remember I talked about the surrogate itself and the anthrax life cycle. Well, we wanted to make a barcoded spore that we could track and trace. In order to develop a good surrogate, you have to recapitulate what is the thing of interest, the properties of interest in Bacillus anthracis. And we were aiming for safety, shape, size, surface characteristics, sample prep. So you have to recapitulate all these things to have a relevant surrogate to test your detection and decontamination equipment. So we selected Bacillus thuringiensis kerstaki because it's 90 years as a biopesticide. There's tons of it made per year. It's used everywhere, and it recapitulates anthracis, or the properties of anthracis, better than BG, the old surrogate. And this gives you a sense of where it's being put down. This is a map of agricultural application of BT, Bacillus thuringiensis. This is a map of where it's been sprayed in Northern Virginia. Here's Dulles Airport. Each of these yellow areas are field applications to control gypsy moths. That raises another problem though, and that is we need to be able to tell ours from everyone else's. So what do? Well, we use synthetic biology to make an artificial genetic signature, a barcode. And so here are a number of applications for this. This technology we can use basically as a novel tool for tracking and predicting. We can use left of boom microbial forensics potentially, where we put these signatures into potential strains that somebody might use, so that it deters that. So there's another possibilities for that. We borrowed the concept from the first synthetic genome that was produced. This is a bacterial genome produced by Craig Venner and his group and Gibson. And this strain contained a number of watermarks to differentiate them from wild type, from the natural thing. And while it was really cool, quotes from Feynman, Oppenheimer, and James Joyce aren't really useful for the biodefense purposes. So we needed something to detect using the methods that we use in the field to detect biological organisms. And we needed something that would recapitulate that all the way from sample prep all the way through PCR detection. So I'll give you a little primer, an overview on PCR itself. PCR is the method that is used in all molecular diagnostic and detection testing. The infamous term that we are all familiar with now from the thing that I promised myself not to mention in this talk. But it involves basically making many copies of a signature, a molecular signature in the pathogen genome. It's make many copies, and that produces a fluorescent signal when you amplify this. And positive test occurs when that fluorescent signal crosses a given threshold. And so we asked ourselves, can we make our own signatures that we can use, and artificial signatures? And the answer to that was yes, we were able to do that. We borrowed some artificial tags from a previous publication, spliced them into the genome, and so they would sit here in the spore, where we would have to get at it through all the methods that you would use to isolate DNA from spores, which is not a trivial thing. When we completed that work, and there's a lot of backstory to that, you can look up in the publications that I posted earlier in the slides. But we then put it out into a real-world testing detection scenario on one of the test fields at Aberdeen Proving Ground. And you can see here a diagram, an aerial view of this. We have a test grid that we put out. Each of these dots here is a sensor or is a filter unit that's pulling in 1,000 liters of air per minute and impacting everything and collecting everything onto a dry filter. And this is this grid. Here we then ran tests, PCR tests on this material, both looking for the barcode itself and looking at for the wild type signal. And you can see here that where the prevailing wind was blowing across the field from this line release, which was right here, you can see that barely on there. You can see that that's where we picked up the signals. We did 2 releases, one of the barcoded strain and one of the untagged strain. And you can see screaming hot sample here in this area, whereas much less sample here, and that's actually better represented here in this case. And so this is the microbial burden that we found. This is the live viable spores that we isolated in each of 2 releases. The barcoded we did first. The wild type we did second, and then the PCR results are down here in looking for both the wild type strain here, the wild type strain, and the barcoded strain. So we see a little bit of a sample here, but this is just barely positive. This is a full-blown positive result here. So the ratio is very different here than here, and we actually think that this is a real signal that comes from bumping off adsorbed material that was collected in the filters or in the sampling units. So, and this is just taken from one of the collectors that was here on the corner, I should mention. So we asked ourselves then, can we make, we did this in a crash program. It was done quickly, as quickly as we could think to do it. And so we asked ourselves, could we do this better? Can we make better signatures that behave, that behave differently? So we wrote an algorithm and a computer program. This is work done by Matt Lux and a number of other folks in the group. And we just put it out on bioRxiv. It's in peer review now, and we expect the results. I expect it back any day on that. So what we did is we designed a program that would basically generate random primer and probe sequences. So we have a primer. These are the primer's annealing sites that allow us to amplify this thing from a very, very, very low copy number into that and generate that fluorescent signal. And then we have the option of using a probe, which itself would generate a fluorescent signal. And that there's a chemistry behind that I won't go into here. But anyway, we were able to show that these don't cross-react with each other. They're actually very specific. And furthermore, none of the 12 that we tested, and this is actually just results for 1, reacted with anything else in this particular panel of potential cross-reacting things, which includes everything up to human DNA as well. So, okay. All right, so that gives you a sense, and the limit of detection is quite sensitive, and we think it's actually lower than 50 copies, but that's really as low as we went in the dilution series. Anyway, so that gives you a flavor of the synthetic, and so what have we done with this? Well, a number of years ago, you probably remember that there was another incident with anthrax spores, live anthrax spores being distributed inadvertently through various through the mail and FedEx or whatever. And the DoD decided that we needed to shore up the science behind how we inactivate anthrax spores by using gamma radiation, which was the standard method that had been done at the time. And so we decided to do this, and we decided to do this in a way that would provide statistical confidence to the method, and that would be replicable. And we heard a lot about in the panel yesterday about the replication crisis. We wanted to be absolutely positive that if you did it, if we put out this method and that we would have the ability to say that if we did it at Edgewood or if we did it at USAMRIID, that we would get the same result in both places. And so we conducted effectively a parallel study where we would take 3 billion spores, we would hit them with increasing doses of gamma irradiation and measure what you'd call the inactivation curve. And you can see in these cases that, pardon me, that the number of viable spores you recover decreases logarithmically with the dose of irradiation that is administered. This is a ton of radiation, by the way. To give you a sense of a human lethal dose is 0.7 grays. We're talking about 42 kilograys, 42,000 grays at the end of this curve. So we're looking at a 1,000,000-fold overkill, and this was reliable in both instances. And so where the barcoded spores come into this was that we needed a standard to judge how effective not only is our irradiation, but how effective is our ability to recover viable spores from the irradiated materials. And it turns out that we then use the viable spores to be able to distinguish any residual live cells if we happen to find them, but also as a recovery standard. You can see that when you're dealing with high density spores, your recovery suffers somewhat when you do it just the spike with no matrix, no dead cells, dead spores in the matrix, versus when you're recovering in 3 batches that we spiked with this material. So high density, and this is a function of density. So you go even higher with the number of dead spores in the matrix, your recovery suffers even more. So you really have to be cognizant of the effects on the matrix. Everything we recovered had the barcode in it, so we're confident that that was good. It was a solid piece of work. Our conclusions from this, and what I want to draw is really can use these barcodes for any number of purposes, and there are people who are studying these and they're using barcodes to study infection dynamics and that sort of thing, and how genetic bottlenecks happen during the infection process. How many, if you seed the lung, for example, with a pathogen, how many actually get further in the infection process and get into the bloodstream, for example. There's been a number of very elegant studies on this. Salmonella really sort of pioneered this a number of years ago. So there's infection progress, genetic bottlenecks in that. We can do what I call left of boom microbial forensics. We've used it to shore up some of the work we've done with the irradiation procedures and quantitate the effects of the matrix on spore recovery, and establish the limits of the limits to which our detection methods for viable spores actually applied. And so this is a very important piece of that particular study. And finally, we actually, and that gets to the use of this as an internal standard. Which brings me to the last slide on this, which is to basically give a plug for work at Edgewood for undergraduate, any interested undergraduate students. We have, and actually high school students in some cases as well. We have a number of internship programs through ORISE, the Army's college qualified leaders and innovative student internship programs, and I have websites here and I can distribute those later. There are also SMART research and engineering scholarships for graduate students who can go to use this to join graduate schools, and then we enroll them into federal civilian service at the completion of their graduate work. And actually, Matt Lux, who I talked about earlier, who developed the genetic barcoding algorithm, was a SMART scholar, and he is now a very successful got one of the people on this SynBio team who is developing synthetic biology approaches to detection. So with that, I will go ahead and go into the Q&A and take questions. So we have some questions here. I've heard the theory that the native population of the Americas was already extremely reduced before the North America was colonized. Okay, so I think I can't really speak to that in terms of the history of that. And I think that there were a number of epidemic waves that swept through as a result of the Columbian introductions. and in South America, and those waves swept north much in the same way that the plague swept north into Europe. I'm not absolutely positive about that, but the evidence for Amherst's involvement in that is clear. That letter is in his hand. It is in his handwriting. He clearly advocated distributing those blankets. So it was obviously the utility was not lost upon them for that kind of thing. So I have a couple other questions. So another question is, I think here we go. I think your mission slide included obscuration. Can you say a little bit about that? Obscuration has been a mission for the, it's one of those missions. I think it fits there with us because it's hard to fit it anywhere else. But the bottom line is on the battlefield, smoke has been used to hide troop movements and things like that for a very, very long time, and it still can be quite useful. And the idea is that we need to have these kinds of materials that are safe, that are not minimally, we want to maximize the safety to anyone who's going to be exposed to that. So that's all I think I really can answer about that off the top of my head, and I hope that does service to your question. There's one more question. I'm not quite clear on it. It says, what's wrong with quotes for your purposes? I'm not sure I understand what the answer to that is. Maybe if you can clarify what you mean by that in the Q&A, I can speak to that a little bit more clearly. But in the meantime, I want to thank everybody for turning out for this. It's been a great opportunity, and I really appreciate your interest in this material. Let's see, is there another? There's one more. Okay, one more question. Oh, okay, yeah. So if the question was about Ira Baldwin's quote regarding the ethics of killing in warfare, that's been a difficult question for humanity to answer since the dawn of warfare, I would imagine. But there have always been considerations of honor, and what is honorable in warfare, what is a dishonorable way. You like the idea of you stab somebody in the back, that's not an honorable way of doing things. And I think that the discomfort with the chemical and biological weapons simply arises from the idea that you don't actually have a direct effect on your enemy. You can't look somebody, it dehumanizes in some way. I think that's been the general feeling on that. And I hope that answers the question. I think we have one more. Oh, the quotes. Yeah, okay. I think if the quotes were about the barcodes, then yeah. So there's nothing wrong with it intrinsically. I think it's very creative, and it certainly speaks to the enterprise and innovation of the thing. For our purposes, though, the signatures are too long. We needed something that really is about 150 nucleotides, and that's it. And only if you think about it. There's a limit to how much information you can actually code into that. So for our purposes, we needed something shorter, and we needed something that would actually be represented, that would mimic a signature that you would look for, that you would be using for actual PCR, which the amplicons, or the length of those signatures, are really only 100 to 150 to 200 nucleotides long. So that's why we decided that the quote approach, however cool it is, and it was not something that we would be pursuing. Does that answer? And again, thanks so much. Really appreciate your time and your comments.